Hi, I am Raina binti Zulkifli. Now I will explain about the principle of chromatography. Chromatography is a technique in which the components of a mixture are separated based on differences in the rate at which they are carried through a fixed or stationary phase by a gaseous or liquid mobile phase. Chromatography is widely used for separations, identifications, and determinations of the chemical component in a complex mixture. Sample is usually a mixture of many components. Sample containing unknown compound. Each of the components must be separated prior to identifications using other analytical method. The intermolecular attractions of each component is different. Some components in the mixture will be more attracted or absorbed towards stationary phase, and some will attract toward mobile phase, and then the component will distribute themselves. Next, I will explain about the column chromatography. In column chromatography, the separations occur on stationary phase that held in a narrow tube where mobile phase is forced through the tube under pressure or by gravity. Stationary phase absorbent in column chromatography in solid phase, and most common use is silica gel or alumina. Mobile phase, pure solvent or a mixture of different solvent in liquid phase. And then the type of separations is column chromatography, solid liquid.
Assalamualaikum warahmatullahi wabarakatuh. My name is Natala Ain binti Onjasmi. In this video, I will explain the application of column chromatography in the pharmaceutical industry. So before we move to the application, we have to know what is liquid chromatography. So liquid chromatography is an analytical technique utilized widely in the pharmaceutical industry. In this industry, our manufactured products need to be of the highest quality to ensure the least risk to patients. It is also to guarantee that goods are certain standards. Researchers, manufacturers, and developers use various technical equipment and analytical technique, including liquid chromatography, during their development process. Not just that, liquid chromatography also used to separate a certain sample into its individual components. The separation occurs when the sample interacts with the mobile and stationary phases. The various parts of the sample are separated out based on their polarities, which they will have varying levels of affinity for the mobile phase, which will result in migration through the column at different speeds. So moving on, the mixed components are placed at the top of the column of the stationary phase, which is generally a fine absorbent solid such as silica. This must be distributed evenly to minimize the presence of air bubbles that could influence the results of the test. The exit of the column is toppered with glass, wool, or a porous plate. When the mobile phase passes through, the mixture separates into bands. These then can be collected and analyzed via other methods. Subsequently, the technique works as the components in a mixture that are attracted to the absorbent surface of the stationary phase with varying degrees, depending on their individual polarity and their unique structural characteristics, which is a component with a high affinity for the stationary phase will migrate down the column slower than a component which has more affinity for the mobile phase. The most common form of liquid chromatography in use today is high-performance liquid chromatography, which is HPLC, which pumps the sample mixture through the column at high pressure.
Assalamualaikum warahmatullahi wabarakatuh, and hi, my name is Nusabrina binti Rahimi, and I'll be talking about how is liquid chromatography used in pharmaceutical. So, how is liquid chromatography used in pharmaceutical? HPLC, or we say as high-performance liquid chromatography, is the form of liquid chromatography that is generally used in the pharmaceutical industry, as it can provide the precise results that are required. The results can be used to analyze finished drug products and their ingredients quantitatively and qualitatively during the manufacturing process. This is achieved through the separation, quantification, and identification of components in a mixture and can be used to reveal the identity of a drug and monitor the progress of a therapy on a disease. So, one of the main benefits of HPLC is its ability to elucidate the structure and determine the quantities of impurities in pharmaceutical formulations. HPLC is especially suitable for compounds that are not easily volatilized thermally and unstable and have high molecular weights. Therefore, it can quantify a drug in its pure and dosage form. Moreover, HPLC may also be used to further our understanding of the normal and disease process in the human body through biomedical and therapeutically research during investigation before of the drugs registrations. The analysis of drugs and metabolites in biological fluids, particularly plasma, serum, or urine, is one of the most demanding, but one of the most common uses of high performance of liquid chromatography. Blood plasma or serum contains numerous endogenous compounds of 10% in concentrations much greater than those of analytes. Analyte concentrations are often low and in the case of drugs, the endogenous compounds are sometimes structurally very similar to the drug to be measured. The binding of drugs to the plasma protein also may occur, which decreases the amount of free compound that is measured. For the application of column chromatography, other than pharmaceutical, it can also use for the forensic chemistry, industrial chemicals, biochemical, and food industry such as antioxidant and additive.
Next, I will continue with the result. For material required, Solution 1 dissolve 40 mg of human insulin in sufficient of 0.01 mHCl to produce 10 ml. For solution 2, mix through 900 microliter of 0.01 mHCl to produce 100 microliter of solution 1. For solution 3, dissolve 40 mg of human insulin EPCRS in sufficient of 0.01 mHCl to produce 10 ml. For solution 4, mix 1 ml of solution 3 with 1 ml of a solution containing 4 mg of processed insulin RS. For mobile phase A, dissolve 28.4 gram of anhydrous Na2SO4 in sufficient water to produce 1000 ml. Add 2.7 ml of orthophosphoric acid. Adjust to pH 2.3 if necessary with ethanol amide. Filter and decant by passing heat through the solution. For mobile phase B, mix 500 ml of mobile phase A with 500 ml of acetonitrile. Filter and decant by passing heat through the solution. Next, with procedure. The HPLC is carried out using a VDEC C18 column of protein and peptides maintained at 400 degrees Celsius as the mobile phase at a flow rate of 1 ml per minute at a mixture of 48 volume of mobile phase A and 55 volume of mobile phase B. Prepare and maintain at a temperature of not less than 20 degrees Celsius and a detection wavelength of 240 nanometer. And this picture show the result. In conclusion, basic chromatography is a technique that allows components of a mixture to be separated based on the variation in how quickly a gaseous or liquid mobile phase transport them through the fixed or stationary phase. Although the pharmaceutical can employ liquid chromatography to provide the lowest danger to the patient, or the manufactured product in the pharmaceutical sector must be of the higher quality. Researchers, producers, and developers employ a variety of technical tools and analytical procedures such as liquid chromatography during the development process to ensure that product meet a particular requirement. That's all for us. Thank you.